Hello, Anatomy and Physiology. This is going to be my last post for a remote learning week. You won't have a bell ringer for tomorrow, just to make sure and get your test done. If you are ahead of the game and want to take your test today, you will be able to. After I'm done making this video and posting, I will go through and email everyone their testing instructions. And so you could take it at some point today if you want, but tomorrow is mostly for that. Today, we are just reviewing a few last minute things. There is a homework assignment to do today, along with the one that I posted yesterday, both of which are questions you're going to see on the test. So make sure and complete both of those assignments before you take your test, and make sure any and all missing work is done and in the grade book before you take your test. I can see your timestamp on both your test and the timestamp when you submit work, and so I'll know if you completed assignments after the test. The whole reason I do that is because those questions are on the test, and you will do better if you take those assignments before the test. I do want you to answer this bell ringer question. Two questions, really. What type of bond forms between amino acids? And then when we do hook a bunch of amino acids together, what do you call that chain of amino acids? Now is the opportunity where you can pause this video before I tell you the answer. Did you actually pause? Okay, maybe you hit play and now you're back, I hope. So this was the structure of our basic amino acid. You can see down here I have alanine as an example. This is just flipped over, kind of upside down compared to the one that we have in our notes here. But you see the hydrogen, central carbon, the amino group, and the carboxyl group. Over here we have our central carbon, the hydrogen. We have our carboxyl group and the amino group. It looks slightly different over here. See how it's CO and it's missing an H, and this one has an extra H on it. This version of alanine has gone through dehydration synthesis, and it's been removed hydrogen on one end, and it kind of shows it more in its basic and amino form. But when we come over to this picture here, this shows the actual process of hooking three separate amino acids together. They're just calling this R1, R2, and R3. It doesn't matter what amino acid they are. It could be alanine, tyrosine, and serine, but this R would change for each of them. But it doesn't matter. We didn't get to do this in class, but had we built these amino acids, I would have had different students. One group would take an oxygen off of their molecule, and then the group sitting next to them would take the two hydrogens off of their amino group, and we would end up forming a covalent bond. But what we call the actual bond that forms between amino acids isn't just, it is a covalent bond, but specifically we call it a peptide bond. And so when we do dehydration synthesis and remove a water and we hook these guys together, so now we're bonding our carboxyl group to the amino group on a neighbor. It would be right here that's happening. Right where the carbon is double bonded to the oxygen. This bond right here would be the peptide bond. Excuse me. This would be the peptide bond. And same thing happened over here, where we removed an oxygen and two hydrogens. This would be our covalent peptide bond. So that's the answer to number one. What's the bond that forms between amino acids? In this case, I have 3 amino acids, but 2 peptide bonds. There's a question on the test about that. So just think, if I have 3 things I want to hook together, I would need to remove just 2 waters in order to get all 3 hooked together on there. So the number of bonds should be 1 less than how many amino acids you have in a chain. If I had 20 amino acids in a chain, there would be 19 peptide bonds in that. Answer number 2 is polypeptide chain. Because we have multiple peptide bonds, we call it polypeptide. And that's what a chain of amino acids is. All right, I hope that helped. You'll see that on the test. I'm not going to go over this with you guys, but just a friendly reminder, if you are working on studying today, I encourage you to fill this out. These little faded out numbers right here, 1, 2, 3, 4, they help answer the secret question or answer whatever down here at the bottom. And so it's like an extra way to double check that all your answers are right. These letters spelled out should form the answer to this question. If you're working on it at home and run into a problem you don't know the answer to, shoot me a message. I'm kind of bored, so let me know if you need help. I wanted to refer back to this page in our notes just to cover one other thing you're going to see on the test that we haven't talked about in class yet in detail, which was the difference between saturated and unsaturated fats. Now, this is a fat and this is a phospholipid. A fat up here can also be fully saturated. The one that's pictured here is saturated because there's no double bonds, and I notice there's no bends in it, no little kinks, if you will, here. So fats can be saturated, or you could have had chains that have bends in it as well. Phospholipids have the same thing going on with their tails as well. But the difference between a saturated and unsaturated fat, which you've probably heard of when we talk about nutrition or just healthy eating and healthy habits, is usually the saturated fats are considered the less healthy of the two. We talk about good fats and bad fats. If you're going to be eating a saturated fat, usually if it's room temperature and solid. So think of butter. You can keep butter out in a butter dish, in your cupboard or on the kitchen table, and it won't melt unless it's crazy hot in your house. They're solid at room temperature. Saturated fats also always come from animal fat. So think Crisco, big tub, or like lard. Those are solid. Bacon grease, okay, once it's cooled down to room temperature, bacon grease turns solid. All of those are considered saturated fats, and they all come from animal products. In that case, saturated fats have no double bonds, and so we use the word they're saturated because they're full of hydrogens because there's no double bonds in there. Unsaturated fats are considered the more healthy option, and they're all plant-based and liquid at room temperature. So think of all the oils: vegetable oil, olive oil, avocado oil, coconut oil. Those are all more healthy, but they're all coming from plant products. And they have at least, if not more, one double bond. They're unsaturated because when there's a double bond, we end up losing a hydrogen on each carbon. And so there's less hydrogens. It's no longer saturated or full of hydrogens. And so that's where the name comes from. So clarifying the differences for you between these two, you will see at least one question on the test about the difference between saturated and unsaturated fats. All right. I also wanted to review with you, because it's on the test, just the basic parts of an atom, but also not just like protons, neutrons, electrons. I know you guys know that, but how you can answer questions about an atom with that background information. So if I'm looking at this picture and I had a periodic table handy, I'm in my classroom and I can see there's one over here, you won't actually need a periodic table for your test. If it makes you feel better to have one handy, you are welcome to, but you don't need one for the test. If you guys were in class, I'd give you the option of taking one if you wanted, but it's not necessary to answer any and all questions on the test. But if I look at this atom right now, I would need to use a periodic table in order to figure out what atom it is. There's one thing I can look at immediately to figure that out. I feel a little like Dora the Explorer here. This is where I'd wait for your response. What's that? You're right. Protons, the number of protons. And I can see just in this image that there are how many protons? There are, yes, good job, 4 protons. And that we have these 4 positives right here. Okay, so 4 protons. Now, when I'm looking at this, my next question is how many neutrons are in this atom? The initial reaction is to want to say 4, but protons and neutrons don't always equal each other. So Notice these are negative. These blue ones here have no charge on them. And I can actually see 1, 2, 3, 4. That's right, 5 neutrons. All right. Now, the number of electrons, negatively charged particles, I have here are 4. With this information, just looking at it, I can tell even from my first question, how many protons are there, what atom it is. When I look at my periodic table, I'm looking for the number 4, and that turns out to be beryllium. Now, so the atomic number is 4. I can also check to see, is this an isotope or not? Here's this question down here. How can I tell if it's an isotope? I would also need the periodic table to answer that question for certainty. But when I look at beryllium, I can see that it has a mass of 9.0122. Round that to 9. What's 4 plus 5? 9. And so this is not an isotope in this case because my mass, my atomic mass units in the center of my nucleus, what makes up the biggest mass of my atom, equals, the standard mass that I find for this atom on Earth. If there were 4 neutrons and I had a periodic table, I could actually say, yes, this is beryllium, but it must be an isotope, because then I would have 8 in here, and that is not equal to the mass number. Hint, because you don't need a periodic table on the test, I'm not going to ask you specifically about an isotope. But if your protons and neutrons do not equal this number, the atomic mass number that's on a periodic table, then you have an isotope. So again, let's say the neutrons were 4, P plus N would be 8. If the mass number is 9, then I know I have an isotope. Now, how could I tell if this is a cation or anion? And remember, a cation has a positive charge and an anion has a negative. Quick, easy way to remember that is look at the little T right here. Think T positive. So a cation is going to be an ion that has a positive charge or negative charge. In this case, I have 4 electrons, 4 neutrons, and so there's no charge to this atom. And so if it was a cation, though, I would have more protons than neutrons, more positives than negatives to make the overall charge positive. And I would know it's an anion if the opposite was true, and that the number of protons was less than the number of electrons, so more negatives than positive. And here's just a little graphic to kind of show you what that might look like. You will see something similar to this on the test. I'm going to give you an atom and ask you if it's a cation, anion, or isotope. What you're going to be looking at is the number of protons, neutrons, and electrons to figure that out. So in this case, this guy, a cation, is positively charged because I have 5 protons and 3 electrons. More protons than electrons. This guy's an anion because I have more negative electrons than positive protons. So it has an overall negative charge. Just a little review for you. Along those same lines, just a quick review about the 3 types of bonds: ionic, covalent, and hydrogen bonds. By the way, these pictures are not in your packet if you're looking for them. We've already done these in your packet, just showing you a different picture to review. So we talk about sodium and chloride all the time, but what makes these an ionic bond is that sodium gives up an electron to chlorine. It then becomes positively charged, a cation, and then chlorine becomes a negatively charged anion, and the attraction between those two is what makes an ionic bond. So you want to pay attention to the charges and how many protons and electrons each atom has if you're trying to decide if it's an ionic bond. A covalent bond, they knit together a little bit more closely. They hug each other. So chlorine gas, Cl2, they actually just share a pair of electrons, and they form a covalent bond when they're sharing electrons. So if you notice that an image or something is showing that they're a little bit more like overlapping or that they're hanging on to electrons with each other, that's a covalent bond. And hydrogen bonds, remember, they form between two different molecules, or within the, like, think of a protein, for example, or DNA. The whole big macromolecule might end up folding back in on itself or forming hydrogen bonds to sort of hold the 3D structure in place. And so it's an attraction between polar areas of an atom. And it doesn't have to be water. DNA is a great example where the nitrogenous bases have polarities to them. And so adenine always pairs with thymine and they form hydrogen bonds. Cytosine and guanine, those are polar with opposite charges and they're attracted to each other always in the same way. And so even in DNA, there are hydrogen bonds, and it has nothing to do with water, but it has to do with molecules having this polarity attraction to each other. It's not covalently bonded. They're not sharing electrons. They're not transferring electrons like ionic. It just has to do with their 3D shape and how they interact. All right. Now, this is what I would recommend doing right before you're ready to take your test. Once I'm done reviewing this with you, I am going to give you instructions on how to log in to your email and access your test. And so stay tuned for that information after we go through and review your concept map. If you're not ready to take your test yet and you still have more work to do, you could pause this video, come back to it later when you are ready. I would do this as soon as possible before you're ready to take the test. So I'm talking to you students who are done all your homework, you're ready to take your test, watch this. All right, this is what we would do in class right before. So Unit 2, basic chemistry, we're going to go over here. We first talked about the atomic structure and our subatomic particles. You all know this. These are the protons, positively charged, neutrons that are neutral, and electrons that are negatively charged. The two particles that you find in the nucleus are your protons and neutrons. They are pretty much the only thing that has atomic mass. So, or at least in great numbers. And so they make up almost the entire mass of your whole atom. The electrons you find out in the electron cloud, and they are responsible for the bonding behavior of each individual type of atom. If you end up with a different number of neutrons than what you would find on the periodic table, we would call it an isotope. So in the example of beryllium, on the periodic table, its atomic mass is 9. And so I would expect to find 4 protons, 5 neutrons in beryllium. If there were 4 protons and 4 neutrons, that means that beryllium has ejected one of the neutrons from its nucleus and now is considered an isotope, perhaps radioactive if it's doing it with great forcefulness. All right, and then we talked about the properties of water. A few key things about that is it has a high specific heat, meaning it avoids temperature change. This is great for you if you walk outside on a freezing day. It is polar. It's a very good solvent. It does all kinds of things because it's polar. Its ability to be attracted or sticky to other things, creep up thin tubes, but it's also a very, very, very good solvent. It cushions and protects your body. Think about your brain's little airbag of cerebral spinal fluid that protects your brain from sloshing around and hitting the inside of your skull. And it is very chemically reactive, particularly when we talk about dehydration, synthesis, as well as hydrolysis. All right, this continues down. I'll go on to the next slide when we talk about pH. But instead, we'll move up here to our different types of chemical bonds. We just reviewed those. But we have ionic, covalent, and hydrogen bonds, of course. Now, we know ionic forms between ions of opposite charges. The covalent bonds, there's 2 types, polar and nonpolar. Polar is when the 2 different sides of the atoms have opposite charges. They're still sharing electrons, but they are sharing them unequally. Think of Mrs. Lloyd's big brother getting to drive the car more often than she did when we were in high school. Big Brother hogged the car more often. We shared the car, just not equally. And so that was more of a polar sharing example. A covalent bond is that's nonpolar, would be sharing electrons perfectly equally. And so the electrons spin, and usually these look symmetrical if you were looking at a diagram of them. Hydrogen bonds, okay, these ones are unique. I mentioned this just previously on the other slide when we were talking about the 3D shape. They are important in water, yes, but also DNA. The two sides of the DNA ladder are held together by hydrogen bonds between the nitrogenous bases. Proteins end up folding and twisting on themselves because of hydrogen bonding, because as you guys learned from your research, some amino acids are polar, some are nonpolar, and the polarity of some amino acids end up making them attracted to other amino acids on a chain. So I might have a long chain of amino acids, and then it starts to bend and fold and twist back on itself till you get a blob-like structure, right, as far as your amino acid goes. And that is all because of hydrogen bonding within the single large 3D molecule, macromolecule itself. All right. Chemical bonds are only, hint over here, involved with electrons. Okay, kind of the same information on both sides here. Electrons are the only particle that's responsible for how an atom reacts and behaves in bonds. And then we talked about types of chemical reactions, synthesis, decomposition, okay? And we didn't really talk about exchange reactions much. These are the ones where the arrows go both ways. And so ATP and ADP, for example, if I add a phosphate to ADP, it turns into ATP. If I remove a phosphate from ATP, it becomes ADP. So that molecule or that reaction can go back and forth. There's no questions on the test about exchange reactions, though. Synthesis reactions, think dehydration, synthesis, same thing, dehydration, synthesis. To be a synthesis reaction, it just means you make something new. In this case, covalent bonds, or if it's between amino acids, we call them peptide bonds. Anytime we are hooking two subunits together or two monomers together to make a polymer, we call that a synthesis reaction. When we remove water, which is most of the time, it's called a dehydration synthesis reaction. Decomposition is the other word for hydrolysis. So something that's going to break things down. In this case, we're going to add water to break particles apart. It's called a decomposition reaction or hydrolysis. All right, and then now we're getting down to our macromolecules down here. So first off, I want to remind you that our most common organic, I shouldn't even say organic, but in all organic chemistry, we are going to be using mostly carbon, hydrogen, oxygen, and nitrogen. The reason I have a little star next to O is that's the most abundant in living things or in organic chemistry. Carbon is very, very important, but it's not the one that we find in greatest numbers. It's oxygen. So little star, that's the big one. Right? I didn't put them in order, although in your notes we did put them in order on page 7. So oxygen comes in at 65%, carbon at 18.5%, hydrogen at 9.5%, and nitrogen at 3.2%. There are other important atoms, but those are the big ones. Oxygen, the biggest, most abundant. All right. Organic catalysts, something that speeds up a reaction that is found in living things, is enzymes. If I ask you a question about which one of these things is an enzyme, if it ends in ACE, A-S-E, you have a enzyme. Okay. So it could be catalase, it could be glucase, lactase, sucrase, oxidase, any of those things. Okay, if it ends in ASE, it's an enzyme. And enzymes are our first type of macromolecule here, proteins. Proteins are very, very, very important, right, in our body. The two types of proteins we discussed in your notes were structural and functional. Enzymes in particular are of the functional variety. their shape has a very, very important job, right? The blob, okay, itself, even though it looks like a blob and looks weird, that shape is very, very key. If it were to denature or change its shape, we would lose the functionality of that protein. And then others are structural. So think hair, okay? These are long, long thins of keratin in here, which is a type of protein that is giving strength and rigidity to even dead cells, okay, to keep them on our head. Things like elastin, okay, that gives some like elasticity to our skin. Some of our cartilages and stuff also contain collagen. Those are structural proteins, things that are firm, that provide support, but they don't do a thing other than providing support and structure in our body and in our cells. Enzymes are functional proteins. Remember that one. All right, coming back down here. Let's keep going with our macromolecules. So then we have lipids. Okay, our lipids, the main three types we talked about were fats, phospholipids, and steroids. Don't forget about steroids. They are a type of lipid. They're nonpolar, like all of this group. Phospholipids are unique because they're kind of half and half. They have a nonpolar side and a polar side, which gives them their unique properties. And then of the fats, you can have a saturated or unsaturated fat. We just reviewed that in the same video. Make sure you know the differences for the test. We also have our carbohydrates. The main groups of carbohydrates were our monosaccharides, disaccharides, and polysaccharides. Make sure to know the examples from your notes for the test. Polysaccharides, in particular, the one I wanted to point out and bring up, like starch, cellulose, and glycogen. Glycogen is unique because we have this in our liver. It's the only thing animals have that is a polysaccharide that we make. Most other polysaccharides we eat, okay? Potato chips, pasta, whatever, all of that stuff usually comes from plants. Most people don't sit down and eat liver to get a super dense carbohydrate meal because most people don't like liver, but you could. Right? Glycogen is in our liver, in animals, and it is the only carbohydrate that we have. It is made up of tiny monosaccharides, glucose in particular. And so glycogen is like the storage form of glucose in our livers for animals. When you are low on sugar, you have hormones that tell your liver to release glycogen, and it's broken down into glucose so you can raise your blood glucose in your body. All right, and then we have nucleic acids. These are your DNA, which I keep hitting this because it's on the test. The sides of the DNA are held together by hydrogen bonds. It helps keep that three-dimensional structure. And then, of course, we have RNA, DNA's helper, and we have ATP, which provides energy for our cells. It's made up of a nucleotide, which is 2 extra phosphates on the end of it. And so it's a nucleic acid as well. We don't oftentimes talk about that in biology. All right, those are your macromolecules. No examples. You should be able to identify a picture of them for the test. That was the bell ringer yesterday. Yeah. All right. And now moving on to the other side, we have coming back down here to our other inorganic things. Acids and bases was another key thing that we discussed. So if you are a proton donor, meaning in water you give up hydrogen ions, you're considered an acid. If you're a proton acceptor, you are a base. And the way we measure our acids and bases is by measuring the amount of hydrogen concentration. We use pH to do that. So neutral is 7. Okay, an acid would be anything below 7, and a base would be anything above 7. Okay, I should have added that on here, but these are below 7 and above 7. The closer a number is to 7, the weaker acid or base it is. So if I said a substance has a pH of 8.6 compared to another substance that's 10.5, and I asked you which one is weaker, you would say the 8.6. That's closer to 7. Also, I could ask you, is that one an acid or a base? and because it's above 7, it would be a base. Make sure you can identify if something's an acid or base based on its number, and also if it's considered weak or not. The closer to 7 it is, the weaker it is. If it's a small number, that's a strong acid, like a 2, a 1, a 0, very, very strong acids. And then the closer to 14 you get, a strong base. And so a 13 would be a very strong base. A 14, crazy strong base. Very, very harsh chemicals usually are those bases. Cleaning supplies, things like that, are typical bases. And then lastly, something in our body that resists pH change, which is important because our pH needs to stay pretty stable. We like to be in homeostasis in our bodies, and those are buffers. Probably the most important buffer is that buffer in our blood that is going to help maintain a pH of slightly basic. Okay. Our blood should be at 7.4, which is a very, very weak base. That's above 7, very close to 7 would be a weak base. Okay, that is it. Please let me know if you have any questions before you take your exam. Now let's talk about how to take your exam. So what you're going to do today when you're ready is go to your email. I'm going to walk you through that. Maybe you already know, okay? You could even fast forward a little bit through this if you want to, but I would go to Great Falls Public Schools website, gfps.k12.mt.us. And from there, I would go to Parents and Students and select Online Resources. On here, this is where you can go to get to Google Drive and all that good stuff. What we are looking for is your webmail. Now, I'm already logged in to my email and webmail. If you don't know how to log into your email, I will attach in Google Classroom this PDF that shows you this. So go to Parents and Students Online Resources, just showed you this. Okay, here it is. And then this has the information about logging in. So you're going to use your regular school district email address. Okay, if your first name was John, your last name was Doe, and you're going to graduate in 2023, john.joe.2023@gfps.k12mt.us. And then it's the regular password that you use to log into Google Drive, when you log into a Chromebook, when you log in anywhere. Okay, and then you might choose no if you're on a school computer. If you're on your personal computer or your phone and you don't have to log in again, go for that. Okay. You should be checking those regularly. One thing students say to me all the time is, Mrs. Lloyd, I can't find your email. I haven't sent them yet. It's 9:36 right now on Thursday. I will be doing that today, this morning, hopefully through this afternoon. But I sent myself one to show you what you're going to get. So it should be coming from me, Amber Lloyd, and it should say Unit 2 test. Let's say you have, like me, okay, oftentimes I get a gazillion Google Classroom things. I clean up my email regularly and delete these announcements. Like, okay, all of these like classroom stuff. Ugh, delete them, get rid of them if you don't need that update and that information. But if you go in Friday night and you have a gazillion posts and you can't find the email, right, you can always just search for my name or search for, let's say, just Unit 2. Okay, and man, I'm searching in my deleted folder here. Okay, it would be in your inbox. Okay, and I would suspect that it would be the first thing in your email. So when you get this email from me, okay, this is kind of what's copied and pasted, but what will be unique for each of you students is your access code. And that's why I have to email each of you individually to give you that access code. So please read this. I know it's long, but I have some important information in here about testing integrity while you are testing from home. And I want you to read it. I want you to take it to heart instead of me just telling you this, because I don't want any misinformation. If you're talking to your friends too, tell them, read the test integrity paragraph. But what you're going to do, okay, here's your step-by-step guide. You're going to go to the student portal, which is linked here for you. And my email should still be open. And you're going to put your student ID number and your access code. I just realized I think I know my access code. Now, we don't have an open submit. Okay? In this case, I want you guys to give me your student ID number. You should be correct. Okay, I gave myself one as a test. Okay, and I hope it works. Nope. I'm going to pause this real quick. Okay, I emailed myself the wrong access code, so I had to go look that up. Okay, this is what makes mistakes all the time. If something doesn't work for you with the login, please let me know. I gave myself the student ID 12345, and then here's my access code that the system automatically generates. And this is why I have to email each of you your code. When you're ready, you're going to say login. This does not start your time, right? Yet, so you can even do this before you're ready to begin, just to make sure it's in there. Now, I assigned myself to all classes, okay, on your guys' case, but in your case, okay, what you guys will be seeing is a start time and end time and a time limit of 60 minutes. Don't click this button until you're ready to begin, but it's going to end at 11:30 p.m. on Friday. The expectation is that you take it sometime today. It's already posted, although you guys don't have access yet, and by 11:30 p.m. on Friday. So you can take it whenever you're ready. Make sure and study, have all your homework in before you do take it. Now, when you do go to take it, okay, I wrote my expectations again in here because I really, really want you guys to read those. My time hasn't started yet, okay, but here's your information. Okay. Additional instructions from your teacher. Okay, you agree to the terms of service. Okay, that you're of age. You guys all are. When you click begin quiz now, this is when your time starts. Okay, and what you will see is a PDF of the test that you'll scroll through. It's 6 pages. This would be like turning the page, and then this is your answer sheet that you would click. All right. You'll only answer 49 questions. Number 50 is a short answer question that you'll reply to the email I sent you with your answer. Okay. And so you can submit once you're all said and done. Okay. I'm going to only answer one question. Let's see what happens. It tells me it hasn't been submitted yet because I haven't answered all of them. It's like, are you sure? I'm going to say yes. I'm not going to get a good grade. Okay. It will tell me, okay, I got 0, the one I guessed on, I did not get right. Okay. Here's your score sheet, but it'll tell you your score right away. Now, I would want to go back to my email, and at the very bottom of this, okay, there is a short answer question. You will just reply to this email and type your answer in right here. So I would say short answer, right, and then begin typing whatever your answer is. The answer is not hot dogs. And then send it along to me, okay, and I'll get that and be able to grade them. I think that's everything, you guys. I hope that you let me know if you have any questions or concerns or you run into any issues at all. I probably will not be online after 3:30 on Friday. I'm going to put my computer away after being on it for so long this week. So if you run into issues on Friday night, Mrs. Lloyd may not be available to help you. I will see you guys all on Monday, and we're all going to be wearing our masks. I know we're all so excited about that. And I miss you. Good luck on your tests. Bye-bye.